Are you looking to pass the biology content knowledge 5235 Praxis 2 test like I am? So currently, I am going to read a segment of the Cliff Notes for the Praxis 2 Biology Content Knowledge 5235. The section that I'm on is Chapter 2, Molecular and Cellular Biology. The section that I'm going to be reading about is biochemical reactions. A biochemical reaction is a process in which chemical bonds between atoms are formed and or broken to produce a new product or products. The starting or raw materials that begin a reaction are called the reactants. The new substance or substances formed as a result of the reaction are called products. The generalized formula for a chemical reaction is as follows. Reactants equals products, or changes into products. Thousands of chemical reactions are taking place in your body right now, converting a variety of reactants into the array of products that maintain your homeostasis, or well-being, such as providing growth and repair of cells, digesting foods, providing energy for movement, and much more. In chemical reactions, energy is absorbed and released. A chemical reaction that releases energy is called an exergamic or exothermic reaction, such as cellular respiration. The products of an exergamic reaction have less energy than the reactants. Chemical reactions that absorb energy from their environment are called endergamic or endothermic reactions. The products of an endergamic reaction have more energy than the reactants, such as photosynthesis. Activation energy is also known as the energy bearer because it presents the amount of energy needed to start a chemical reaction. The activation energy needed to start a biochemical reaction is usually in the form of heat or chemical energy. Biological catalysts or enzymes lower the amount of activation energy needed to start a reaction. Enzymes are mostly proteins and catalyze most of the biochemical reactions in your body. Enzymes speed up the rate of biochemical reactions that would either not occur without enzyme catalysts or would occur at rates too slow to support life. The following list details 5 common types of biochemical reactions. Synthesis reactions. A biochemical reaction that forms a product from 2 or more substances. A plus B turns into C. Photosynthesis is a synthesis reaction that combines water and carbon dioxide to make carbohydrates in an endergamic reaction that requires photons of light energy to overcome the activation energy and power of the reaction. Decomposition reactions. A reaction in which a large molecule is broken down into smaller molecules. C turns into A plus B. An example of a decomposition reaction is the breakdown of foods into component molecules, such as the decomposition of table sugar into glucose and fructose. Decomposition reactions are the opposite of synthesis reactions. Oxidation or combustion reactions. Oxygen combines with other substances in a variety of ways. Cellular respiration is a slow exergamic oxidation of food to release stored energy. Food plus oxygen turns into energy. ATP plus waste products, water and carbon dioxide. Neutralization reaction. A neutralization reaction occurs when an acid and base of equal strength and volume combine to form a salt and water solution with a neutral pH. Acid plus base turns into salt plus water. On occasion, you may take an antacid that neutralizes stomach acid to relieve an upset stomach. Condensation reactions. Condensation reactions are a type of synthesis reaction. 2 smaller organic molecules combine to form a more complex molecule, releasing a molecule of water in the process. Carbohydrate subunits or monomers are chemically bonded in a series of condensation reactions that release water and form large carbohydrate molecules. Example, glucose molecules combine to form starch in plants or glycogen in animals. pH. pH is the negative algorithm of the hydrogen ion H+ concentration. The pH scale measures the acidity or alkalinity of a solution. Acidic solutions have a pH range of 0 up to 7. The lower the number, the stronger the acid. Basic or alkaline solutions have a pH range greater than 7 to 14. The higher the number, the stronger the base. A pH of 7 is considered neutral. An acid, such as hydrochloric acid, is a chemical that releases hydrogen ions when dissolved in water. Bases like sodium hydroxide release hydroxide ions when dissolved in the water. The pH of a solution can be changed by adding more acid or more base. Equal volumes of equal concentrations of acids and bases neutralize each other. Note that buffers may limit the ability of acids and bases to change pH. Buffers. A buffer is a special type of solution that resists change in pH when an acid or base is added. Chemically, a buffer is a solution containing a weak acid and its conjugate base. Acids are neutralized by bases when the positive hydrogen ions react with the weak base to form water. Bases are neutralized when the sodium hydroxide ions released with the positive hydrogen atom in the weak acid to form water. Buffers work by absorbing or reacting with the positive hydrogen or negative sodium hydroxide ions to form water. Some biologically important buffers include phosphate, bicarbonate, and protein, which resist changes in the pH of biological fluids. Biologically important molecules. Living organisms are mostly composed of organic compounds and water. Recall that organic compounds contain carbon. Organic molecules are involved in every biochemical reaction that occurs in every living organism. Interestingly, all biochemical reactions must take place in water. 4 biologically important organic compounds include proteins, carbohydrates, lipids, and nucleic acids. Another biologically significant inorganic compound is water. Proteins and enzymes. Proteins are organic polymers that are made by joining amino acid monomers together in a specific order. Properly functioning proteins exist as a folded three-dimensional structure. Changes in temperature, pH, pressure, salinity, free radicals, or exposure to excessive radioactivity may result in changes in the three-dimensional structure of a protein. Changes in the three-dimensional structure of a protein alter the function of a protein, and extreme changes in the structure lead to denaturation, which is the complete loss of 3D structure. Surprising, only 20 amino acids function as the organic monomers for all proteins. Amino acids have the 4 bonds of a central carbon atom connected to an amino group, a carboxyl group, a hydrogen atom, and a side chain of variable complexity. Observe the amino acid components in the following graphic. So they demonstrated out, carbon is in the middle, and then there are 4 branches out to R, which is considered a side chain of variable complexity, COOH, which is a carboxyl group. It also has an H, which is a hydrogen atom, and it has an NH2, and NH2 is standing for the connected amino group. Amino acids combined as a condensation reaction, forming a peptide bond between the amino group of one amino acid and the carboxyl group of another amino acid with the release of water. Peptides are formed when amino acids are linked together. Dipeptides are a linkage of 2 amino acids. Polypeptides are multiple linkages. There are 4 types of biologically significant proteins: enzymes, transport proteins, structure proteins, and hormones. An enzyme is a specialized reusable protein that speeds up a metabolic reaction without being changed. Enzymes are biological catalysts that lower the activation energy for a reaction. How are enzymes so specific in the reactions they catalyze? An active site on the enzyme chemically binds to a unique receptor site on the substrate, forming an enzyme-substrate complex. The specificity of the enzyme-substrate complex is often referred to as the lock and key. The enzyme-substrate complex lowers the activation energy for the reaction to be catalyzed. Enzyme names generally indicate what they catalyze and end with the suffix of ase. For instance, the enzyme maltase catalyzes the reaction where maltose is broken down into two glucose molecules. Some enzymes require non-protein cofactors to bind with the active site to facilitate the binding with the substrate. Inorganic cofactors include metal ions, like iron. Organic cofactors are commonly called coenzymes and include vitamins. Prosthetic groups are cofactors that are chemically bonded by the protein to the protein. Enzyme-substrate reactions can be prevented by materials that act as inhibitors. Inhibitors are substances that attach to the enzyme, thereby preventing the catalyzing of a reaction. There are two basic types of inhibitors, non-competitive and competitive. Heavy metals are poisonous because they are non-competitive inhibitors that attach to the enzyme. Non-competitive inhibitors bind at sites other than the active site, but then they force the active site to change shape. Competitive inhibitors bind at the active site and physically block the substrate from binding. They are typically homologous to the normal substrate and prevent enzyme functions. Transport proteins move smaller molecules through the body and through cell membranes. Hemoglobin is a transport protein found in red blood cells that carries oxygen to cells. Structural proteins form materials that cells use for structural purposes, such as proteins of the cytoskeleton. Some structural proteins give cells their shape. Collagen and keratin are types of structural proteins that make up skin, fur, feathers, wool, fingernails, hooves, ligaments, tendons, and parts of the bone. Hormones are chemical messengers that are made in one gland but travel via bloodstream to trigger a response in another part of the body. Some hormones are protein or partly protein, such as insulin. An overabundance of sugar in the blood stimulates the release of insulin from the islets of the Langerhans in the pancreas. Insulin triggers the uptake of blood glucose by the liver, muscles, and fat tissues. Diabetes mellitus results when the pancreas does not secrete any insulin, type 1 diabetes, or when the pancreas secretes an insufficient amount of insulin, or the body is resistant to the insulin produced, type 2 diabetes. The four groups of hormones, based on structure, are amino acid derivatives, peptides, steroids, and isocyanates. Carbohydrates. Carbohydrates are the most important source of energy for your body. Sugars and starches are common carbohydrates. Your body digests carbohydrates into glucose that fuel the cellular respiration reactions that release energy as needed to the cells in your body. Excess glucose is stored in glycogen in your liver and muscles. Carbohydrates are easily identifiable because they are made only from carbon, hydrogen, and oxygen. Carbohydrates are hydrates of carbon, which makes remembering their general formula easier. Carbon times H2O. The ratio of carbon to hydrogen and oxygen is always 1 to 2 to 1 in a hydrate of carbon. Carbohydrates contain multiple hydroxyl groups and carbonyl functioning groups. Carbohydrates range in size from a single monomer like glucose to large polymers made from thousands of monomers like cellulose. Carbohydrates are divided into three classes based on their complexity: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are often called the simple sugars. The simplest carbon, glucose, is a monosaccharide and is often given intravenously to hospital patients. Other common examples of monosaccharides include galactose and fructose. Fructose, also known as fruit sugar, because it is the most common carbohydrate in most fruits. Glucose and fructose are isomers because they both have the same formulas, C6H12O6, but different atom arrangements. Glucose and galactose are stereoisomers because the hydrogen and hydroxyl groups are orientated differently around one carbon atom. Notice that monosaccharides have a carbonyl group, CH2OH, on one carbon atom and a hydroxyl group, and hydroxyl groups most of the other carbon atoms. When monosaccharides are in a solution, they exist in open chains and closed ring structures, as seen in this previous graphic. The cylindric closed ring structures are more stable at equilibrium. Disaccharides are formed when two monosaccharides are chemically bonded together in a condensation reaction that liberates a molecule of water. The most common disaccharide is sucrose, also recognized as common table sugar. Sucrose is formed by the condensation between the monosaccharides fructose and glucose. Lactose, or milk sugar, is formed by the condensation of glucose and galactose. Polysaccharides are complex carbohydrates that are polymers formed by the condensation reactions linking many monosaccharides. Glycogen is a common polysaccharide that is composed of glucose subunits. Glycogen functions are stored energy and is kept in the liver and muscles of humans and other animals. Starch and cellulose are common polysaccharides composed of glucose and made by plants. Starch is a water-insoluble molecule used to store energy in plants, or glycogen in animals. Cellulose is also water-insoluble, but is used as a structural component for forming rigid plant cell walls, such as the cell walls found in wood. Glycogen, starch, and cellulose are composed of glucose monosaccharides, but the chemical bonds between them are orientated differently. Therefore, humans can ingest glycogen and starch, but not cellulose. The cellulose that you consume in fruits and vegetables is indigestible and scours through your digestive system as dietary fiber or roughage. Lipids. Lipids are large nonpolar organic molecules composed of carbon, hydrogen, and oxygen. Lipids are unlike carbohydrates because the ratio of hydrogen to oxygen is always greater than 2 to 1. Also, lipids are not polymers. Lipids have four major functions. They store energy long-term in fats. They provide structure to cell membranes as phospholipids. They act as a water barrier, waxes, and they help various body functions maintain homeostasis, steroids, some vitamins, and hormones. Triglycerides are a type of lipid formed when three fatty acid molecules bond with a glycerol molecule. Fatty acids are long chain, 12 to 20 carbon atoms. Carboxylic acids with a general formula of RCOOH. Saturated fatty acids have no double bonds, but instead are saturated with hydrogen atoms. Unsaturated fatty acids have one or more double bonds. Triglycerides can be either a solid, fats, or liquid oils. Note the components of a triglyceride in the graphic below. When energy molecules are abundant, fat cells store the excess energy as triglycerides. Enzymes in the cells break down triglycerides when energy is required. Interesting, soap is made in a process called sponification. A strong base is added to triglycerides to form molecules that have a polar charged end and a nonpolar end. The nonpolar end bonds with the dirt and oils, while the polar end is the water soluble and rises the entire mess away. A typical cell membrane is a phospholipid bilayer, in which the nonpolar tails of the fatty acid point inward and the polar heads point to the outside surface. This lipid bilayer acts as a barrier, allowing the cell to regulate what enters and exits through the membrane. Waxes are lipids that are formed by combining a fatty acid with a long-chain alcohol. Plants and animals make waxes to prevent water loss, for protection, and for structural purposes. Earwax catches harmful products and protects the inner ear. The wax coating on some plants' leaves prevents water loss, and honeybees make wax to build honeycombs. Steroids are lipids that are made of multiple closed ring structures and do not contain fatty acid chains. The sex hormones, cholesterol, and vitamin D are common examples of steroids. The sex hormone estrogen is made in the ovaries. Estrogen controls the development of secondary sex characteristics in females and regulates the menstrual cycle. Testosterone is synthesized in the testes and controls the development of secondary sex characteristics in males, such as facial hair, muscle mass, and deepening voice. Sadly, the use of anabolic steroids by athletes to increase muscle mass for performance enhanced is well documented. That's where we're going to end. I hope that you do well on your test. I'm hoping to do well on mine. Good luck.